It's only been a short time.
And the probably green mice. You said that when you got them back to the States.
That was a little joke, actually. We didn't, they were for sale in a marketplace in Thailand. They're dyed, of course. I mean, it's like chicks at Easter. You know, once the feathers grow out, they're no longer the color they once were.
I'm actually going to make part statement, but also part question for you. A little bit of time. Clearly, this is work that only NIH or in general NIH would be supportive of, since it's very basic. But can you take us through how long from when they started looking at this science to where we got to where there's sort of a breakthrough in understanding the coronavirus and the XMRV?
Well, I mean, research on retroviruses in particular, as a significant focus of NIH research goes back to the 60s. I was a graduate student with Howard Temin, who was one of the co-discoverers of reverse transcriptase, which was discovered basically how retroviruses are different from everything else and how they replicate. He was one of the sort of very early scientists studying. So I'm in kind of the second generation of people who've been, whose work has been supported by NIH to study these. Studying of retroviruses over the years, not only there's a number of important things. I think this is one of them, but it's not one of the ones I'm going to list. One is it really led to the basic groundwork for understanding cancer. None of the other systems that were being looked at the time really provided the insight into cancer causation that retroviruses did. And that's because I didn't talk about this at all, but retroviruses can pick up genes called oncogenes into the virion and then pass them on from one cell to another in an experimental setting. And because there are only 3 or 4 genes in a retrovirus, an oncogene is only 1 of 3 genes. In a cancer cell, it's 1 of 20,000 genes. You can't tell which one it is in any easy way. In a virus, it's easy to see it there, and it's easy to find it. It's easy to eventually to work out what it does. And that's why Mike Bishop won the Nobel Prize, for example, was for doing exactly that. And Harold Varmus. And the other thing is that all of that research laid very important groundwork so that we could move very rapidly when the AIDS epidemic struck, identify HIV, develop tests for it, and begin to understand how it worked and develop antiretroviral drugs, which have been extremely important. If we didn't have antiretroviral drugs in the U.S. now, I can't imagine what it would be like there. You'd probably be, it'd be awful. It's just the way people were infected with HIV would be treated and so on and so forth. And the way they still are, of course, in Africa, where antiretroviral drugs are not very common. And this kind of research, much of it extremely basic. I mean, we're talking about viruses in chickens and mice originally, causing cancer in chickens and mice, not in humans. None of this early work was done in humans until the 80s, until 15 years or more after the work had been supported. Was there really any research in human retroviruses? And then, of course, the AIDS epidemic burst on the scene. So the research that was supported by NIH for all that time, really, without that, I can't imagine where we would be in our understanding of these diseases and the development of treatments and tests and so on and so forth. In parallel, of course, there's been an enormous body of other kinds of science that have contributed to this. The development of genome sequencing, whole genome sequencing, for example, is moving very fast. It'll be possible. It's almost possible now to go into a doctor's office, have a sample of DNA taken, and get your genome sequenced for diagnostic purposes. We're not quite there. It's not quite to that, but I think it will be. That will be a diagnostic modality for some things within 5 years or so. And all of that, of course, contributes very much to our ability to be able to find viruses in DNA. We have the sequences of the DNA where we can go digging through by computer. So instead of digging up fossils, with a pick and the hard hat, and we dig with a computer and going poke through the genome of the computer to find these things. Thank you very much.